Okay, so good morning or good afternoon, everyone, and welcome. I'm Rihanna. I'm an ambassador of Trinidad and Tobago and a director of biochemistry at the Youth Team Advisors. And this weekend, we'll be learning about microbiology as per the agenda of the Biological Sciences Research Program. So I hope you guys are excited to learn a lot about microbiology, and who knows, maybe this will help you pick a future career path. Maybe you want to study microbiology after this. I hope, because it's really cool. So let's get into the lesson. Oh, I skipped a little bit too far. Sorry about that. So we have some objectives before we start. So just a general outline of how today's lessons is going to run. So firstly, we have the introduction, which is the importance of microbiomes and host pathogen biology. Secondly, we have microbiology basics, so structure, classifications, metabolism, and quorum sensing. Third, we have host pathogen interplay with immunity, antibodies, MHC, and evasion strategies. And fourth, we have applied cases with microbiomes and health, so our gut-brain axis and antibiotic resistance. Right, and before we begin, let's do something fun. So I have a tiny icebreaker for you all. So I'd like each of you to introduce yourself with one word that describes what you expect to learn today and one adjective that captures your current mood. So tell me your expectations basically for the workshop using one word, and then introduce yourself with an adjective that describes your current mood. So right now, I'm pretty hungry. I really want some fries right now, so I would say hungry Rihanna. Or maybe right now you're sad Rihanna, or happy Rihanna, or something like that, right? So you're probably wondering what kind of weird icebreaker this girl wants me to say right now. Well, it's more than just an icebreaker, right, guys? It's a reminder to us that science is personal. Our moods, our expectations, even our health are tied to these invisible communities of microbiomes living in and around us. So, without further ado, let's get into the introduction. So, I want you guys to think for a few seconds, what do you think microbiology is? I'm gonna give you guys a couple of seconds to think about it, and then I'll explain. So, what is microbiology? Microbiology refers to the scientific study of microscopic organisms that cannot be viewed using the naked eye. So, for example, bacteria, viruses, fungi, protozoa, and some algae. Microbiologists in particular, they research these organisms to gain knowledge about their structure, functions, genetics, metabolism, the way they interact with their environments, and human beings as well. So you're probably wondering, well, that's cool and all, but why should I care about this? How does this concern me? Well, simply put, microbiomes affect nearly every aspect of our life. They affect our health, diseases, industries, environments. They make people sick. They make organisms sick. They may be helpful, such as in the gut or in fermentation, and also how they interact with each other and ecosystems is very important for nutrient recycling, decomposition of waste, and even to clean up oil spills in some case. So they're really our allies and our enemies in a way, microbes that is. Sometimes even at the same time, they're our allies and our enemies. So understanding them is the first step towards controlling them. So we're going to go into specification and classification. And we're going to start with the prokaryotic world, bacteria, of course. So bacteria is a single-celled organism without a nucleus. And well, their simplicity actually hides their incredible adaptability. So take E. coli, for example. Most strains live harmlessly in our gut, helping us digest food. But some, like E. coli, 0157 causes severe food poisoning and kidney failure. Same species, completely different genes, completely different outcome, right? Bacteria can come in three main shapes. There are spherical, rods, and spirals, to be put simply. The actual scientific names are a little bit more complicated, so I think just knowing the shape for you guys should be a little more simple. And well, you can see it right underneath there. You can try to pronounce it as well too. Nothing's stopping you. So their cell wall composition lets us classify them as gram-positive or gram-negative. And this distinction is not just academic. It determines which antibiotics work. For instance, gram-negative bacteria like Pseudomonas aeruginosa, oh my god, I always pronounce this one wrong. Aeruginosa, I think. Pseudomonas aeruginosa. I hope I'm saying it right. I never say it right. So, for instance, gram-negative bacteria such as that, they are notoriously resistant to many drugs. So they really help us identify which types of antibiotics actually work. And for plasmids as well too, those little circles of DNA often carry antibiotic resistant genes, meaning a harmless bacterium can suddenly become untreatable after acquiring one. I know, right? That's really, really scary. So now we're going to move on to the hijackers. Guess what? They are viruses. So I hope you guys saw these things. I'll just leave it up for a little bit longer so you could just catch the slides a little bit, because I moved a little bit fast there, so I apologize for that. So now we're going to move on to those hijackers, which are viruses. So viruses aren't even alive by some definitions. They're genetic material wrapped in protein, sometimes with a lipid envelope stolen from the host. So take HIV. It inserts its genetic material into host DNA, turning human cells into virus factories. Or influenza, which changes its surface proteins so frequently that we need a new vaccine every single year. And of course, SARS, which caused the COVID-19 pandemic. A microscopic particle less than 0.1 micrometers in size just brought the entire world to a standstill. Economies shut down, health systems collapsed, and over 6 million lives were lost. That is the power of viruses. We're going to move on to our friends and foes, which would be fungi or fungi, whichever you guys like to call it, because you know you're a fun guy. Okay. Okay, that was a lame joke. Okay, let's continue, and we're going to ignore that lame joke. So fungi, they are basically eukaryotes, meaning they have a nuclei and organelles just like us. So they can be helpful. For example, yeast, because yeast, they give us bread, beer, and wine. And some molds like penicillium give us antibiotics. But fungi can also be dangerous. There's a specific fungi, I believe it's Candida albicans, that normally lives harmlessly in our bodies, but in immunocompromised patients, it could cause life-threatening infections. So there's another fungi, Aspergillus fumigatus, I believe, which causes lung infections in people with weakened immune systems. So, for example, a real-world example of this was in 2022, actually. There was an outbreak of Candida aureus, which is a multi-drug resistant fungus that spread in hospitals worldwide. And it can survive on surfaces resistant to most antifungal drugs and cause deadly bloodstream infections. That's why fungi is just as urgent as bacteria and viruses. Understanding fungi is just as urgent as bacteria and viruses. So now that we have a general understanding of our specification and classification of bacteria, viruses, and fungi, we can move on to microbial metabolism. So that's basically a fancier way of how microbes power themselves, right? So I'm gonna pause for a few seconds. You guys could pause the video as well to the recording to take a read, because everything is quite important. So I want you guys to read. You don't need to process it right now. I'll explain. Just read and I will explain. I'm gonna give you guys 2 seconds. Okay, so microbes, sorry, why do I keep saying microbiomes? Microbes generate energy in 3 main ways. The first one would be aerobic respiration, second, anaerobic respiration, and third, fermentation. So we're going to touch briefly on aerobic respiration, right? So aerobic metabolism is the metabolism in which glucose is completely broken down by the use of oxygen as a source of energy from microbiomes. So there's something called glycolysis. That's the starting point of the Krebs cycle, and the electron transport chain is involved in this. And oxygen is the final electron acceptor. So in a very simple way to put it, aerobic respiration utilizes oxygen and it's highly efficient. So for example, in E. coli, again, in the gut where oxygen is available, aerobic respiration occurs where it's highly efficient. And this process, again, is very efficient in yielding the greatest quantity of adenosine triphosphate, which is ATP or energy per glucose molecule. And seeing that oxygen is available because aerobic requires oxygen, many bacteria, such as E. coli, again, switch to aerobic metabolism because it maximizes energy gain. So in other words, certain microbes are able to generate energy through aerobic metabolism because it's very efficient. Because of that use of oxygen in this entire metabolism process, bacteria such as E. coli are able to survive in the gut when oxygen is available. They're able to maximize energy gain because oxygen as a final acceptor causes a very high adenosine triphosphate yield or ATP yield because it's so efficient. And now we have anaerobic metabolism. So when there is no oxygen, a situation referred to as anaerobic metabolism occurs, which involves anaerobic respiration. Other molecules like nitrate, sulfate, and carbon dioxide replace oxygen as an acceptor of electrons used by microbes. So we will have less energy than that of aerobic respiration, but more than fermentation. So this renders microbes capable of surviving in an oxygen-deficient medium, whether this be in soil, in sediment, or in gastrointestinal tracts. So let's think about this realistically in the world, right? We see bacteria popping up in the weirdest places. It doesn't need to be somewhere where they have oxygen. They always pop up at such a weird place. That's because of the fact that they don't need oxygen to survive. They could respire anaerobically. They require no oxygen at all for anaerobic respiration. So they could survive in these very oxygen deficient environments and spread. So for example, Clostridium, that Clostridium species in soil and the gut, they don't have much oxygen available to them, yet they are still prevalent because of the fact that they use anaerobic metabolism, right? And lastly, the third type of microbial metabolism would be fermentation. So fermentation produces the least amount of energy, but it still allows some kind of survival, right? So when microbes are not able to access oxygen or alternative acceptors, fermentation is used. They are unable to use oxidative molar and produce lactic acid. And lactic acid is something that's produced when, let's just say, you're exercising. It's gonna lactic acid is going to build up in your body, and that's what causes your muscle aches and stuff like that. And it's cured by inhaling more oxygen to kind of level it out in a simple way. So produce lactic acid, ethanol, or carbon dioxide to recycle NAD5, incorporating only glycolysis. Fermentation does not even master much ATP per glucose because it bypasses both the Krebs cycle and the electron transport chain. But nevertheless, it's needed to survive and has real-life applications, such as making bread, yogurts, and alcohols. So this is why yeast can ferment sugar into alcohol and CO2 to make beer and bread, right? So these processes, this particular process, I would say, isn't just biology. They're also industry, because fermentation is worth billions of dollars in food and beverage production. So without microbial metabolism, we'd have no yogurt, we'd have no wine, we'd have no cheese, and we have no antibiotics either. And I don't know about you guys, but I really love cheese. So if this didn't exist, I feel like I would have been deprived of my favorite food. So now that we've touched on aerobic, anaerobic, and fermentation, microbial metabolism, we could move into quorum sensing. And quorum sensing is a little bit complex, which is why, again, I'm going to allow you guys to pause, explain it very simply, and then we move on, right? So, bacterial communication, or in other words, the social networks of bacteria, that would be quorum sensing. So, bacteria don't live alone, they communicate. Quorum sensing is how they send chemical signals to coordinate group behavior. So, for example, Vibrio fischeri, a marine bacterium, only glows when enough bacteria is present, helping squid camouflage in the ocean at night. So, many bacteria species are capable of communicating with one another to organize group behavior, and that process is referred to as quorum sensing. It enables the bacteria to detect their population by means of chemical messages known as autoinducers. So like we see with the bacteria that helps squids camouflage in the ocean at night, right? At a particular population density, a high degree of gene expression is mutually induced by bacteria. This coordinated action enables the population to behave as a multicellular being in a certain sense. So obviously they need to work together, and multicellular organisms, all the cells there work together to help living organisms survive. Main and most common example, humans, right? So while that is all well and good, there is something we need to know about quorum sensing. It could be a little bit dark as well too sometimes. It plays a darker role as well, right? So remember we mentioned earlier on in bacteria, I believe it was the Pseudomonas aeruginosa. Again, still can't pronounce that. I've tried, guys. They use quorum sensing actually to form biofilms in the lungs of cystic fibrosis patients, and that makes their infections nearly impossible to treat. So that was a little fun fact for you guys, because I don't know, I just find that so incredibly insane, you know? So quorum sensing is through signals in the production, release, and detection of signals, right? So in the gram-negative, so bacteria, the most common ones are the Achillohomoserine lactones, whilst in the gram-positive, oligopeptides are used. Sorry guys, my glasses. I need my glasses. So as the bacterial colony increases, the autoinducers increase in the surrounding. Because obviously, as population density increases, and we learned this in ecology as well yesterday, density of a population affects the amount of consumers, producers, predators that are present. So as that bacterial colony increases, the autoinducers also increase in the surroundings. And after critical concentration, these molecules attach to receptor proteins and they trigger transcription of target genes, and that may provoke a response to produce virulence factors to form biofilms, engage in bioluminescence, or other such collective action that benefits from bacterial population. So again, what all these big words mean in a simple sense. Again, an example of the Vibrio fischeri, which is a marine bacterium like we discussed before. So if you remember just now, I said that it only glows when enough bacteria is present. So they help squid camouflage in the ocean at night. So that's what they mean in the sense of engaging in bioluminescence. That's a big example of it as well too. So quorum sensing within itself, it's significant in pathogenic bacteria as well too. Again, we discussed that example earlier on. I was really excited about talking about this, so I kind of mentioned those two examples before I was supposed to talk about it, but we'll talk about it again. So another example is in Pseudomonas aeruginosa. So they also use quorum sensing to form a biofilm of bacteria in the lungs of patients with cystic fibrosis, which could make their infections nearly impossible to treat. And again, Vibrio fischeri glows via quorum sensing to develop a bioluminescent glow in its symbiotic relationship with squid of some species. So not all species, right? Because like we learned yesterday with ecology, there are different types of ecological relationships, and this bacteria will only be able to have a mutual or commensalistic relationship with some of them, right? So other than pathogenesis, quorum sensing has other valuable applications in the field of environmental microbiology and biotechnology, as scientists seek out ways in which they can utilize this form of communication to their advantage. And this research has huge implications. Imagine new drugs that silence bacterial conversations instead of killing them, preventing infection without even driving resistance. Wouldn't that be insane? Your white blood cells wouldn't even have to act, they just do it for you. Imagine that, right? But let's not imagine too hard, because then we might come a little bit too delusional, but it can happen. It can happen. So now that we know about bacterial communication, this is just a small diagram, and you guys could look at this you guys could look at this and pause the video to see it more clearly after listening to what I said about this. And we're going to move on now to host pathogen interactions. Don't worry guys, we're almost done. So we're going to start with the first line of defense. In other words, we're going to start with innate immunity. So the first line of defense is innate immunity and responds quickly but without particularity. So it bears our physical barriers. So the skin and mucous membranes in our nose, the defenses applied by cells such as macrophages, neutrophils, and natural killer cells or NK cells, as well as those soluble barriers we see that complements proteins and interferons. So think of them as security guards. They check on everyone, but they don't remember everybody's faces. They attack broadly but without much memory. Innate immunity is initiated by recognition of a common microbial characteristics. So they are pathogen associated molecular patterns, right? So in terms of pattern recognition, receptors like TAL-like receptors, they could identify them sort of then, but not completely, because if we think about it, the skin can't really identify what kind of microbacteria is coming into our body, right? The skin just serves as a first line of defense to prevent any sorts of bacteria from entering our body. And although it is efficient to kill most pathogens in a short period of time, innate immunity is not memory based. It does not evolve to kill certain microbiomes. So you know that we have memory lymphocytes, I believe. We have memory cells in our immune system which will remember how certain diseases and viruses would have acted and entered into your body. So they kind of just target and kill those viruses and sicknesses before they have the chance to start again. But with innate immunity, we don't have that. So like I said again, there are security guards who check everyone, but they don't remember faces. So they attack broadly, but without any memory. So, and then we have adaptive immunity. So adaptive immunity, those are the specialists. We finally reached the second floor of the hospital with more of the specialists. So adaptive immunity is slower to respond, but initially has a highly specific target and has memory for future infections. So it's slow but precise. So for example, B cells make antibodies that target specific invaders. T cells either kill infected cells or coordinate their immune response, and once they're exposed, they remember. So like adaptive immunity is kind of like that one girl that is just hold a grudge against you for some reason. I don't really know why, but she always knows why, and she just knows how you act and everything, you know. And this is also why vaccines work. So for example, after surviving chickenpox as a child, right, most people never get it again because our body knows exactly what this disease has done to us and how to combat it before it gets worse. That's adaptive memory in action, guys. So to summarize that, innate is rapid, but it's non-specific and they have no memory. Adaptive is initially slower, but it's highly specific and we have long-term memory. Now we're going to talk about immune evasion. Well, I include some stuff about antibodies here, but now that I think about it, these are things that you guys will have to more learn off in terms of functions in particular. So we could just take a read about it. This is kind of very advanced, so I just wanted to mention it to you guys so you would know exactly what certain things do in your body. So we could just touch on it. So antibodies, why do I keep saying that? Antibodies are proteins produced by B cells and they recognize and neutralize specific antigens. So there are 5 main types: IgM, IgG, IgA, IgE, and IgD. So IgM is the first antibody produced during infection. IgG is long-term immunity and crosses the placenta. IgA is found in mucosal surfaces, so for example your gut and respiratory tract. IgE mediates allergic responses and defense against parasites, and IgD is involved in B cell activation. And remember, we talked about B cells here. That's what produces antibodies, and T cells are helper cells in the sense that they kind of handle that immune response and they coordinate immune response, and they either kill infected cells as well too. So major histocompatibility complex, or MHC, those molecules are protein on cell surfaces that present antigens to T cells. So there are 2 MHCs. So MHC1, it presents antigens from inside the cell to the CD8 plus toxic T cells. MHC2 presents external antigens to the CD4 plus helper T cells. So I know it's very advanced, which is why I didn't really want to touch on it that much. I just wanted you guys to have an idea of what it does. So don't worry if you don't understand it that much, because it is a lot more higher and advanced. So I think you guys, as like a little homework, y'all could research into your favorite antibody here, which one you think sounds more interesting after that their little like introduction. So if you want to do IgM, you research what it does, or if you want to do T cells or B cells, cytotoxic T cells, CD8, CD4, it's really up to you. I don't want to overwhelm you with so much of those antibodies, MHC or T and B cells, right? It's just small little things that you guys could know, right? So, but that little bold text there, those are more important, right? So antibodies neutralize or mark pathogens. That's the first point we're going to take away from this part of the lesson. 2 would be MHC molecules present antigens to T cells, and T and B cells are central to adaptive immunity. That's the main 3 things that you have to know with regards to antibodies, MHC, and T or B cells, right? Don't get too flustered thinking about the rest. Okay, long text. I know, kind of long. Not kind of, it is a little bit long. But so I'm gonna give you guys a few seconds to just pause and read, and then I'm gonna summarize it in quite literally a few sentences, because we don't want it to be too long of a lesson and go over time as well too. So pathogens have managed to come up with sophisticated mechanisms to avoid or overcome host immune systems, enabling them to survive, replicate, and sometimes cause chronic infections. In others, surface proteins are changed continually, a process known as antigenic variation, which makes it impossible for the immune system to recognize it as an influenza virus. Others hide in the cells of a host or interfere with essential immune systems such as MHC molecules so as to prevent detection by T cells. Bacteria are able to enter biofilms, making them difficult to attack by the immune system, and some of the pathogens can produce molecular mimicry to appear similar to the host molecules and not get recognized. Such strategies and tactics increase the difficulty to eradicate infections, and they cause serious problems to cures and vaccine development. In other words, I know that was real long to explain something so simple. They are basically tricking us. They're like the pathogens playbook in a sense. They're tricksters. They fight back, and we don't even realize it. Our body doesn't detect it. So, for example, the flu virus, right? It constantly changes its surface proteins so that it is not detected. And it enters your body and gets to a point where you detect it a little bit too late and you get very sick. HIV, it hides inside host DNA. That's what we call latency as well too. Some bacteria like Streptococcus pyogenes, I believe that's how you pronounce it, they mimic human molecules as well too, like we talked about molecular mimicry. Others like Staphylococcus aureus form biofilms shielding themselves from attack. This is the reason why so much diseases remain so difficult to cure, because they're acting like they are in a play and they're playing every single character in a play. They keep changing their appearance, they keep changing their outfits and doing these quick costume changes on stage so that we don't detect them. We don't realize that it's the same person playing 25 different characters. That's why they're so difficult to detect. So finally, I know you guys are fed up of hearing me yap, so I'm gonna keep this relatively short and we're gonna go into the case studies So we just have two case studies, which is the gut-brain axis, and then we have antibiotic resistance, right? So human microbiome and health. So human microbiome refers to the trillions of microorganisms inhabiting the body in and on it. So bacteria, viruses, fungi, and microorganisms. And these microorganisms become extremely important in the process of digestion, vitamin synthesis, building the immune system, and warding off harmful pathogens. A favorable microbiome is a pillar of good health, whereas imbalance has the potential to lead us to such conditions like obesity, allergies, autoimmune diseases, and infections. So the main thing that we need to focus on is the microbes that live in the gut, skin, mouth, and other body sites, their function, which is to support digestion, nutrient synthesis, and immunity, and the fact that imbalance has the potential to lead to terrible health conditions. So we're going to go into apply the case study 1, the gut-brain axis. So there's a simple way to explain the gut-brain axis, and I'm going to do the simple way, right? So the human microbiome is a vast ecosystem. Trillions of microbes living on and inside us, right? In the gut, they produce neurotransmitters like serotonin, dopamine, and short-chain fatty acids. And these molecules travel via the vagus nerve, an immune system, to the brain. That's why gut health affects mental health. There's a direct link between your gut health and your mental health. So studies show that imbalances in that microbiome or dysbiosis, which is what they call microbiome imbalances, are linked to things like anxiety, depression, autism, and even Parkinson's disease, which is crazy. And a striking example of this would be patients with severe Clostridium difficile infection, and they often undergo fecal microbiota transplantation. Actually, that is crazy, isn't it? So by restoring healthy gut bacteria, not only does the infection resolve, but some patients report improvement in mood and cognition. That is the gut-brain axis in action. You need to take care of your gut by feeding it things like fiber, enough water, enough nutrients and proteins to make sure that that gut microbiome is healthy enough so that your mental health doesn't suffer. Now the second case study, which deals with antibiotic resistance mechanisms. So antibiotic resistance is actually one of the greatest threats to modern medicine. So take MRSA for example. Once confined to hospitals, it's now found in communities and is resistant to multiple drugs. And in the US alone, MRSA kills around 10,000 people per year. That's a huge number of people, guys. Another example is the NDM1 bacteria, first discovered in India. They produce an enzyme that destroys almost all known antibiotics. Almost all known antibiotics. Let that sink in. Infections with NDM1 bacteria are nearly untreatable. So the World Health Organization warns that if resistance continues, routine surgeries like hip replacements or C-sections would become deadly again, because simple infections won't respond to treatment. And the alarming truth is resistance spreads quickly, like Flash and Superman combined, very quickly. Bacteria, they share resistance genes through horizontal gene transfer, even across species. It's like a cheat code that everyone could use, which is why finishing prescribed antibiotics is crucial and why global policies are being pushed to limit unnecessarily using in humans and agriculture. Sorry, that was a stupid way to put that sentence. The alarming truth about it is that resistance spreads quickly. So because they share their genes through horizontal gene transfer across species, it's like a cheat code that everyone can use, right? And finishing prescribed antibiotics is crucial. And that's why global policies are being pushed to limit unnecessary use in humans and agriculture. Sorry, I fumbled over my words there. I got too passionate talking about antibiotic resistance mechanisms. But everything is here for you guys to read and understand as well too. I explained it in a very simple way so that you guys could grasp it properly and that you wouldn't really have much problems after when you go through it again and you listen to what I said and you read, you will understand it more. Because microbiology is a very heavy topic and not everyone will understand it immediately, right? So, finally, conclusion, yay! So, to conclude, microbes are everywhere. They are helpers, they are enemies, maybe both. They digest our food, protect us from disease, and produce medicines. But they could also cause pandemics, resist drugs, and threaten our survival. Microbiology in itself isn't just about science. It's about the future of medicine, food, and the environment. By understanding microbiomes and host pathogen interactions, we learn not only how to fight diseases, but how to live in harmony with the invisible organisms that shape our world. So, thank you very much. That was the end of our lesson. So now I'll be taking any questions that you guys would have. So what part of today's microscopic world did surprise you the most? I would love to hear as well too. And I hope the lesson was sort of light. I hope you learned something. I hope I didn't confuse anyone as well too. And a huge thanks to the youth team advisors for having such an amazing biological science research program. I know you guys have been enjoying it. And with any